Good afternoon, dear students. Now we are going to continue with bacteriological analysis of water. See, in the last lectures, I told you about how to collect and handle sample, how sample is collected and handled, and what are the different, indicators of fecal pollution that we have discussed. Now today we are going to discuss various tests that are used for bacteriological analysis of water. Now what are the various tests? Now bacteriological analysis of water mainly includes standard plate count, or which is a standard procedure for quantitative determination of bacteria in water, that is SPC. So heterotrophic count, even various bacterial counts can be done by standard plate method. But in your syllabus we have to discuss only about presence of or detecting the coliforms and fecal streptococci in water. So we are going to directly switch over to the second test, that is test for presence of coliform bacteria and fecal streptococci in water. So bacteriological analysis of water includes various tests. For quantitative determination we go for standard plate count method. If we want to test the presence of fecal coliforms or fecal streptococci, then we perform multiple tube dilution technique, also called as multiple tube fermentation technique, for fecal coliforms as well as for fecal streptococci, and membrane filter technique MFT for fecal coliforms and for fecal streptococci. Now today we are going to discuss multiple tube dilution technique or multiple tube fermentation technique for detecting the presence of coliforms in water. Okay. So now the multiple tube dilution technique for detection of coliforms in water consists of three tests. Number 1, presumptive test. Number 2, confirmed test. And number 3, completed test. Okay. Now, if we look at the protocol of the presumptive, confirmed and completed test, in case of presumptive test, the medium that is used is either lactose broth or McConkey's broth or lauryl tryptose broth. Any of the three medium can be used. Okay, and in actual practice we have to take 3 sets. Each set contains 5 tubes. That means 3 sets of 15 test tubes. Okay, so there are 5 tubes in each set. So 3 sets comprise of 15 tubes. And in the first set of 5 tubes we take double strength broth. In the second set of 5 tubes we take single strength broth, that is 9 ml, and in the third set of 5 tubes we take 9.9 ml of single strength broth. Which broth? Either lactose broth or McConkey's broth or lauryl tryptose broth. The medium contains indicator whose color change will tell us whether organism has produced acid from lactose or not. In each of the tube we place a Durham's tube for checking gas production by the microorganism. Now all the tubes, that is all 15 tubes containing the medium, are sterilized, and after sterilization we have to add water sample into the tubes. In the first set of 5 tubes we add 10 ml of water sample. In the second set of 5 tubes we add 1 ml of water sample, and in the third set of 5 tubes we add 0.1 ml of water sample, and incubate all the tubes at 35 degrees Celsius for 24 hours and check for gas production. Now if acid and gas is produced in any of the 15 tubes, then presumptive test is supposed to be positive. If there is no acid and gas production in any tube after 24 hours of incubation, you further incubate all the 15 tubes for next 24 hours. Okay. And then check acid and gas production after completion of 48 hours. Now if any one of the tubes shows acid and gas production after 48 hours of incubation, presumptive test is said to be positive. And if there is no acid and gas production even after incubation of 48 hours, then presumptive test is negative and coliforms are absent. Okay. Now after presumptive test we go for confirmed test. Why do we need to do confirmed test? Okay. Why there is a need of performing confirmed test? See, presumptive test I told you. Okay. Now, why do we need to confirm the test? If presumptive test comes out to be positive, you presume that coliforms are present. But then why confirmed test? This is because sometimes a positive presumptive test may be false. That means we get false positive presumptive test. Okay, a positive presumptive test does not necessarily mean that coliform organisms are present. False positive presumptive test is also expected. Why? Because of the following reasons. Because of the presence of aerobic or facultatively anaerobic spore forming gas producing bacteria. Coliforms are non-spore forming bacteria. Okay. So, such organisms may be present. The gas production may be because of synergistic action, synergistic fermentation of lactose by gas producing symbionts. There may be seasonal occurrence of coliform organism not surviving the detection procedure employed. Then because of antagonism also, or because of presence of oxidase positive bacteria capable of producing gas from lactose. For example, Bacillus, Serratia, Irvinia, etc. So, to know whether, we just want to confirm whether the presumptive test that is obtained positive is because of coliforms only. So, we have to perform confirmed test. Okay. Now, but how can we eliminate this false positive presumptive test? The false positive presumptive test can be eliminated by incorporating suitable dyes, that is triphenyl methane dyes, in the fermentation medium. The triphenyl methane dyes, when incorporated into the medium, inhibit the growth of non-coliform organism and will thus prevent the production of gas by non-coliform bacteria. Okay. So anyway, once we get presumptive test positive, we have to go for confirmed test. Now, all the positive presumptive tubes are subjected to confirmed test. Okay. How to do confirmed test? For confirmed test, we use specific medias, selective medias. Okay. What are the selective medias that can be used? Brilliant green lactose bile broth. Right? The other two medias are Endogar or Embiagar can be used. Endogar or Embiagar is taken in plates. Brilliant green lactose bile broth is taken in test tube with a Durham's tube inserted inside the brilliant green lactose bile broth. In short, we call it BGLBB. Okay. Now, a loopful of culture from positive presumptive tube is inoculated into brilliant green lactose bile broth and incubate at 35 to 37 degrees Celsius for 24 hours and check for gas production. If gas production is observed in brilliant green lactose bile broth, coliforms are present. And if gas production does not occur in brilliant green lactose bile broth, coliforms are absent. Okay. If you get gas production in BGLBB, then loopful of culture from this positive BGLBB is inoculated on the surface of, it is streaked on the surface of EMB or Endogar, and look for the presence of typical colonies or atypical colonies. Now, a loopful of culture from positive presumptive tube is streaked on the surface of EMB or Endogar. Incubate the plates at 37 degrees Celsius for 24 hours, 35 to 37 degrees Celsius for 24 hours, and check for the appearance of colonies. Now see whether it is Embiagar or Endogar, three different types of colonies can be obtained. Number one, typical colonies produced by fecal coliforms, that is E. coli. Atypical colonies produced by non-fecal coliforms. And number three, negative colonies. Okay. Now what are negative colonies? Colonies which are colorless, which do not have any color, pale. They are produced by non-coliform bacteria. When negative colonies are produced, means no colorless colonies or pale colonies are produced by microbes. That means the coliforms are absent. Okay. Now, if you get typical colonies, it indicates that coliforms are present. What are typical colonies? Purple nucleated, or you can say purple black centered colonies with or without green metallic sheen are produced on Embiagar. Dark purple colonies with golden metallic sheen are produced on the surface of Endogar. Okay, so this purple or black centered colonies with or without metallic sheen are called as typical colonies, which are produced by fecal coliforms. Okay, atypical colonies are opaque, non-nucleated, pink in color. Atypical colonies are opaque, non-nucleated, pink in color, produced by non-fecal coliforms. So whether we get typical or atypical colonies, it indicates that coliforms are present. They may be fecal or they may be non-fecal. Okay, but if we get typical colonies with metallic sheen, purple dark centered or black centered, it is because of fecal coliforms. Once you get confirmed test positive, you have to complete the test. Okay, so now let us discuss about confirmed test in detail. Okay, now what is brilliant green lactose bile broth? It's a selective medium, especially used for the detection of coliforms. It favors the growth of only coliforms by inhibiting others. Okay, so we look full of culture from positive presumptive tube is inoculated into BGLVB, incubated and check for the presence of gas production. If gas is produced, coliforms are present. Brilliant green lactose bile broth contains brilliant green. It is a triphenyl methane dye which inhibits the growth of non-coliform bacteria. Gram-positive bacteria are inhibited. Okay, so brilliant green is not an indicator, it's a dye. It inhibits the growth of non-coliform as well as gram-positive bacteria. The bile salt which is present in the medium favors the growth of coliforms, and hence BGLBB is a selective medium, selectively allowing the growth of only coliforms. Okay, so gas production in BGLBB indicates presence of coliforms. Endogar is again a differential medium used for the detection as well as confirmation of coliforms. It is prepared by adding basic fuchsine previously decolorized with sodium sulfite to a melted lactose agar base. That means the medium contains lactose, sodium sulfite and basic fuchsine. But basic fuchsine is previously decolorized. Basic fuchsine is a mixture of 3 different dyes: para-rosanilin, rosanilin and magenta 2. Now, let us explain this by using an example of para-rosanilin. It is initially present in, I can say, colorless, decolorized condition. So, this is a para-rosanilin quininoid salt which is colored compound. Okay, it reacts with sulfite or it gets combined with sulfite and forms a non-quininoid compound which is colorless. So, basic fuchsine which contains para-rosanilin, for example, is present in the medium previously in decolorized condition upon combination with sulfite that is present in the medium. When you inoculate it with the positive presumptive tube, okay, 3 different types of colonies can be obtained, as I told you. Purple dark centered colonies with golden metallic sheen are produced by E. coli. Atypical colonies are all pink in color, and negative colonies are colorless, produced by other organisms. It is believed that Endogar is a trapping medium for acetaldehyde which is responsible for giving different colored colonies. When is acetaldehyde produced? Acetaldehyde is produced as an intermediate compound during the fermentation of lactose. E. coli or coliforms are lactose fermenting organisms. They ferment lactose and acetaldehyde is produced as an intermediate product. This acetaldehyde reacts with sodium sulfite, okay, which is present in combination with sulfite, which is present in combination with decolorized basic fuchsine and forms an addition product. Because of that, because acetaldehyde combines with sulfite group, okay, present in the decolorized basic fuchsine, the basic fuchsine is released from the combination. Okay, this releases basic fuchsine from the combination and it gives agar a deep red color. Okay, so we get a deep purple or deep red colonies, rather deep purple colonies. The golden metallic sheen that is produced on the surface of colonies on Endogar is due to precipitation of the liberated stain. Okay, it is due to the precipitation of the stain, that is basic fuchsine, which is liberated in proportional to acetaldehyde that is formed during the lactose fermentation. Okay, presence of at least one typical colony confirms the presence of coliforms. Right. On Embiagar, why do we get typical, atypical colonies and all? It's again a differential medium as well as a selective medium used in confirmed test. It is prepared by adding definite quantities of stains, namely eosin and methylene blue, to a melted lactose agar base. It contains 2 stains, eosin and methylene blue, added to a melted lactose agar base. When we inoculate it with the culture, 3 different types of colonies are obtained. Typical purple dark centered colonies with or without metallic sheen produced by fecal coliforms. Atypical colonies, pink in color, opaque, produced by non-fecal coliforms, rather coliforms only, and negative colonies are produced by non-coliform bacteria. The color of the colonies on Embiagar depends on 2 factors. Number 1, reaction of eosin with methylene blue, that is acidic stain with basic stain, to form a acidic or basic neutral compound. The formation of sufficient acid by lactose fermenting organism which changes the pH of the medium Okay, when the pH of the medium goes towards acidic side, the stains are liberated. The stain is taken up by the organism, that is by the colonies. In this way, E. coli will produce typical colonies and other coliforms develop pink colored atypical colonies. Okay, so that is about confirmed test. Now, after performing confirmed test, we have to go for completed test. Whatever may be the case, whether you get typical colonies or atypical colonies, sometimes some coliforms fail to produce typical colonies. So whether you get typical or atypical colonies, whether they are pink in color or purple dark nucleated, you have to complete the test. How to complete the test? The test is completed by performing gram staining and motility. Okay, so typical pink colored colonies or pink colored colonies present on the medium or purple dark centered colonies present on the medium, that is endo or Embiagar, is streaked on a nutrient agar slant, and small amount of colony is inoculated into lactose broth tube containing Durham's tube. Okay, now the organism will grow on the surface of nutrient agar slant. So the nutrient agar slant culture is used to perform gram staining, and the growth which is present in lactose broth is used to check the motility. At the same time, you can also check acid and gas production in lactose broth. When you perform gram staining and motility, and if you get gram negative short rods, they are coliforms. And gram negative short rods which are sluggishly motile definitely indicate the presence of coliforms in water. This is how we complete the test. Okay. So in completed test, what we do? Typical colony from endo or Embiagar is transferred to nutrient agar slant and lactose broth and incubated at 35 degrees Celsius for 24 hours. In lactose broth, you check for gas production and acid production and go for motility. A nutrient agar slant culture, the growth is used for gram staining. So you should get gram negative short rods which are sluggishly motile. This completes the test. And if you get gram negative short rods and sluggishly motile organism, it indicates that coliforms are present in the sample. Okay, right. So what is the interpretation? If typical colonies appear on the plate after 24 hours, confirmed test is positive, coliforms are present. But if only atypical colonies are produced, the confirmed test cannot be considered negative, since some coliforms fail to produce typical colonies. And hence, regardless of whether you get typical or atypical colonies, it is necessary to complete the test. How do we complete the test? By performing gram staining and motility. Okay. And what is the gram nature of coliforms? They are gram negative short rods. Okay. So you complete the test, and presence of gram negative short rods which are sluggishly motile confirms the presence of coliform organisms. Right. This is about multiple tube dilution technique for coliforms. It is one of the bacteriological analysis done for testing. Okay, it is one of the tests used in bacteriological analysis of organism. The others are membrane filter technique for coliforms, MTDT for fecal streptococci, MFT for fecal streptococci, that we will be studying in the coming lectures. Okay, that is all for today.